Hello, everyone, and welcome to the first of the UHN Foundation's three-part series, Krembil Discovery Livestream Series. Today's broadcast showcases the pioneering research at the Krembil Brain Institute. I'm Christian Cote, host of Behind the Breakthrough, the podcast all about groundbreaking medical research and the people behind it at the University Health Network. We're so glad you could join us and hope you are staying safe and taking care of each other as we begin to see the light at the end of the tunnel of this COVID-19 pandemic. As valued members of our community, we're so pleased to be hosting these virtual presentations to provide you an exclusive insight into a number of the amazing scientific innovations and discoveries happening at UHN every day. This Krembil Discovery Series is inspired by the generous donors to the 2020 Discovery Ball. That was, of course, intended to be a celebratory in-person event, but public health and safety guidelines suggested otherwise. So, disappointing, yes, but in an incredible show of support, our sponsors and donors stepped up and honored their donation commitments. That really is a testimony to the unwavering belief in the research ongoing at the Krembil Research Institute. So, thanks to their generosity, these funds will go towards the core research budget of the Krembil Research Institute, which supports all three of the institutes we're highlighting in this series. Today, we'll be talking one-on-one with the co-directors of the Krembil Brain Institute, Dr. Don Weaver and Dr. Gelareh Zadeh, about their vision as well as their groundbreaking research. We're also going to introduce you to some of the rising research stars at the Krembil Brain Institute, and we want you to be a part of today's conversation. So, as the live stream unfolds, if a question comes to mind, we want to hear from you. Go to the Slido page. The link's at the bottom of the page. You type in the hashtag, or type in hashtag 424160. That's hashtag 424160, and you can submit a question. As well, you can upvote questions you want to hear answered, and we'll get to them a little later in the broadcast. One programming note, please keep in mind for privacy reasons, we won't be able to field anything of a personal medical nature. Now, for those of you who may be unfamiliar with the three pillars of research at the Krembil Research Institute, our communications team at the UHN Foundation has produced this fabulous short sizzle reel to bring you up to speed.
Diseases of the brain, joints, and eyes impact millions around the world, more of us every day. Most medications and treatments address symptoms. What we need are cures. The brain is the most complex and least understood organ in our body. Researchers at the Krembil Brain Institute are not only unraveling its mysteries, they're on the cusp of game-changing breakthroughs to halt the progression of dementia and Alzheimer's, to treat complex brain tumors, stop seizures, diagnose Parkinson's and other movement disorders earlier and with more accuracy, to help people walk again, stop stroke damage, and even prevent strokes from happening. In other words, they're using their brains to save yours. By the time arthritis is diagnosed, the damage is often done. The Schroeder Arthritis Institute is decoding the complexities of arthritis to build a cure. Here, researchers are developing blood tests to diagnose it early, predicting outcomes of surgical and medical treatments to provide ultra-precise and personalized care, and working on world-first therapies to treat, halt, and even reverse the damage. Their ultimate mission? To cure arthritis in our lifetime. Diseases of the eye impact people of all ages and stages, and they're on the rise. The Donald K. Johnson Eye Institute is gaining new and extraordinary insights into these diseases to slow vision loss and help people see again. They're working to restore eyesight through cell transplantation, using 3D printing and developing a drug to treat glaucoma. They're exploring how virtual reality can help retrain the eyes of people with vision loss, and working on a drug that could save eye cells from dying. It's not just about giving people back their sight, it's about giving them back their lives too. For researchers at UHN's Toronto Western Hospital, relentless is more than just a word. It's a driving force. After all, we're not just here to study these diseases, we're here to stop them cold.
Well, from that, it's clear the relentless pursuit of new treatments and cures is it really in the DNA of the Krembil Research Institute, where scientists and physicians are working tirelessly to eradicate brain-related conditions such as Parkinson's, Alzheimer's, epilepsy, stroke, concussion, and pain. And critical to nurturing that talent really is leadership. Dr. Don Weaver is the director of the Krembil Research Institute and research co-director to the Krembil Brain Institute. Among his many distinctions and extraordinary achievements, Dr. Weaver is the only qualified person in Canada to be a practicing neurologist with a PhD in medicinal chemistry and drug design. He is a world-leading researcher in the search to cure Alzheimer's and dementia. Welcome, Dr. Weaver.
And thank you. I would like to thank everyone who has taken time out today on this lovely summer afternoon to join us. I'm just going to try to put things into perspective. As you are probably aware, UHN is a relatively complicated place. There has individual hospitals at different sites. I like to think of UHN as consisting of the Toronto Western Hospital on Bathurst, and then a couple of other places up on University Avenue. Princess Margaret and Toronto General, I think their names are. Each one of these individual sites has a research institute associated with it. So the Krembil Research Institute is the research institute that is associated with the Toronto Western site. And at the Krembil Research Institute, we are absolutely passionate and focused on taking on the diseases that chronically affect people and shorten their lives, make their lives difficult. If we think of what are the three leading causes for why people end up in chronic care facilities and long-term care sites, they are neurological, arthritis, and vision. They have strokes or dementia or some cause which results in their no longer being able to live at home. They have arthritis that is so bad they can't manage on their own, or due to visual difficulties, they cannot manage on their own. And those are the three leading causes, and those are the three areas that we take on at the Krembil Research Institute through our three areas, through the Krembil Brain Institute, the Schroeder Arthritis Institute, and the Johnson Eye Institute. So we really are focusing on all of these three important areas. As Christian mentioned at the outset, today is dedicated and focused to the Krembil Brain Institute. And so at the Krembil Brain Institute, this is where we're looking at the chronic neurologic diseases that really have a huge impact, not only on patients, but their families and indeed society as a whole. So I hope that you will enjoy the presentations that we have today as we give a highlight of just some of the research that we have ongoing at the Krembil Brain Institute. And we have the name Krembil for a very important reason. Through the name Krembil, we acknowledge the exceptionally generous and pioneering support of the Krembil Foundation and the Krembil family in enabling us to build this wonderful institute and to do much of the groundbreaking research that is happening at our location.
Thanks, Dr. Weaver. We're going to see you later in the show, but before I let you go, we'd be remiss in not talking about your own recent developments in terms of pioneering research. And I know you have a trial, an upcoming phase 1 trial. Yep. Of your own that will be of particular interest to patients and families with Alzheimer's disease. Do you mind sharing a little bit about the scope of that trial happening?
Certainly Alzheimer's disease is in the news a lot lately, particularly in the last 24 hours. There are multiple approaches to treating Alzheimer's disease, and our lab is focused on a variety of these, but one of these is on a neuroinflammation. And I like to think of COVID, actually, the pandemic that's happening now, as the other pandemic, because Alzheimer's is a pandemic in its own right and was here devastating individuals long before COVID. And Alzheimer's and COVID share a lot of similarities. They are disorders in which inflammation plays a huge role. And so in part of our work in designing compounds and discovering compounds, we noted that a common drug called furosemide has really impressive anti-inflammatory activities. And we actually have an ongoing clinical trial right now at multiple sites in Canada and outside Canada as well on inhaled furosemide as a treatment for COVID. Now, furosemide itself doesn't get into the brain and therefore cannot be used for Alzheimer's disease, but we are working on analogs of this and we're bringing these along as a potential therapeutic for the treatment of Alzheimer's. So we stay committed and focused on the development of drugs for Alzheimer's disease.
And Don, can I just ask, do you have a sense in terms of timeline for conclusion of this this phase of the trial and then, of course, public?
Shall I say, that's in the control of people who are running the clinical trial, and they never move as fast as I wanted to.
Fair enough. Okay, let's bring our co-director of the Krembil Brain Institute, Dr. Gelareh Zadeh, a leader who inspires by example and action. Listen to this. In addition to being co-director, Dr. Zadeh is also medical director of the Krembil Brain Institute. She is head of the Division of Neurosurgery in UHN's Sprott Department of Surgery, the Wilkins Family Chair in Neurosurgical Brain Tumor Research, and a senior scientist at the Krembil Research Institute. Dr. Zadeh also holds the Dan Family Chair in Neurosurgery at UHN and U of T. And that's an appointment that makes her the first woman ever to hold a neurosurgery chair in Canada. Welcome, Dr. Zadeh.
Thank you, Christian. Thanks for the kind introduction.
Listen, you earned it. Let's start high level. Talk to us about your priorities at the Krembil Brain Institute.
Sure. And again, thank you to everybody who's tuned in. It's a huge opportunity for us to have the ability to speak with you about Krembil Brain Institute. As Dr. Weaver mentioned, one of our key broad priorities and strategies is to integrate research into clinical practice and rapidly translate findings into care delivery as much as possible. And we are one of the few centers that has the breadth and depth of the different disciplines of neurosciences, whether it's in neurology, neurosurgery, neuroradiology, neuropathology, neurointensive care, and neuroanesthesia. And in my opinion, our strength is that with a group of strong neuroscientists, together with our clinicians, we sit under one roof and can introduce innovations in discoveries and technologies in approaches for care for patients. And so as an extension of that, if we synthesize down, what are some of our priorities on short-term basis? For example, we had a key priority to introduce advanced neuroimaging, and we built the Slate Advanced Neuroimaging Center, where we've been able to upgrade the technologies that we use for magnetic resonance imaging, or MR imaging, to the state of the art. And we are able to recruit scientists and clinicians that help us, in fact, integrate all of the advanced imaging that we need to better understand diseases that affect the brain and the spinal cord and peripheral nerves. The other area that I would say is currently one of our key priorities in short period to be able to put together is to establish a cutting-edge, world-class clinical trials research unit where we can, in fact, conduct the many trials, for example, that Dr. Weaver mentioned earlier, on-site locally and be able to test the various therapeutics that we offer for our patients, and also be able to integrate our care in the various disciplines in the form of clinical trials. And then another area that I would say is really critical for us is to modernize the spaces that we have, primarily for our patient experience, so that it becomes a smooth transition from one specialty to the other and allows bringing everybody together in one place to be able to have a fulsome assessment for the patient in a very modernized space where the information that we gather from, again, brain, spine, and nerve function can in fact be captured and integrated into using machine learning algorithms, artificial intelligence, to help improve the care of the patients from the moment they hit the door of the hospital to the completion of their care.
That's terrific. Thanks, Gelareh. Let's pivot to your research because you're pioneering new ground with regard to a really aggressive cancer in the brain called glioblastoma, which is a tumor. Now, your surgical specialty is going in while the patient is awake to try and excise or reduce the tumor, but it's often intertwined with nerves that control various motor functions in the body. So that's delicate. I understand, though, during the surgery, you take a sample of the glioblastoma to study in the lab. So what are you looking for when you examine these samples?
So actually, for all of our tumors, we do a multiple number of analysis, and currently we use almost all genomic and epigenomic platforms to better understand the biology of the tumor, to integrate that result in a multiple platform manner to be able to really appreciate all of the aberrations, all of the changes that happen at single gene level and at global genetic level, to be able to then better understand how disease behaves and be able to identify therapeutic targets. An area that we've really made an impact, and we are one of the very few centers in the world that uses this technique to refine classification, and as a consequence, the decisions we make for our patients, is using methylation. Methylation classification allows us to change a diagnosis in a more accurate manner by approximately 15 to 20%. It also allows us, in very complex cases, up to 80% to make a change in the management decisions that we provide the patients. We're a center of referral. Well, I would say comfortably we're one of the 4, if not fewer, centers in the world that now does methylation to a substantial level. The other area that we've been able to leverage is to use these methylation signatures and look at patients' plasma or blood and see whether the same signatures can be detected in the blood. And in fact, last year we published on this. We're able to accurately detect whether somebody has a brain tumor or not based on a blood test. Of course, you need the MRI to show that there is a tumor there, but the subtype of the tumor, which could be a number of different types of cancers that we get in the brain, the subtype of it can be refined using the blood test. And that really is an example where the world of science integrates into clinical practice, and you can provide a transformation in how we treat patients.
And let's just drill down a bit on the blood test, because that's a breakthrough of yours. That, I mean, talk to our audience about the fact that it's way better for the patient, right? Because typically, prior to your blood test discovery, to be able to diagnose glioblastoma involved surgery.
Yes.
So talk to us about the benefit for patients now with this blood test.
Right, Christian. I think we have to do the next step, which is to demonstrate that this can be a clinical grade test, that the accuracy is high enough that we can, in fact, replace this with what we do currently, which is to do an invasive procedure and obtain tissue for diagnosis. It can also be used to then be able to predict the outcome for patients and refine our decision as to whether we need to operate in the first place or not. Ultimately, I can also see that this type of test, which relies on methylation in the plasma, can be applied not just to tumors, but in fact degenerative conditions. So as Don mentioned, it could be applied to dementia, to Alzheimer's, and other forms of neurodegenerative conditions that impact the brain. And if we can demonstrate that, and we are the center that should be able to, in fact, accurately implement this, we will then transform how we manage patients in our diagnosis of neurodegenerative conditions and be able to detect disease earlier so that we can intervene faster and sooner. One of the biggest challenges for us in understanding brain disease conditions is that often the diagnosis is made mid to late stage of the disease, and interventions are not preventative, but rather interventions are intended to prevent worsening. If we're able to detect disease, diagnose early, and use it as a screening tool, the interventions will hopefully become more effective, and we can then implement strategies that would prevent deterioration and further degeneration.
That's amazing. Okay, before I bring in some of the research fellows that we want to feature today, Gelareh, can you maybe walk us through some of the other pioneering work that's ongoing at the Krembil Brain Institute? I was told to ask you about Dr. Andreas Lozano's high-frequency liquid biopsies.
Yes, so I mean, you're putting me in a difficult position, Christian, because there's so many to select from, but we have a few minutes. So just because you brought up this example, the high-frequency ultrasound is a technology that is absolutely cutting edge. We have a very well-established program at the Krembil Brain Institute where the high-frequency ultrasound in a non-invasive manner can treat disease and conditions of the brain such as tremor, Parkinson's, and other. And so that same technology can also be applied to treat brain tumors by opening up the blood-brain barrier, by being able to increase the amount of chemotherapeutics that's delivered into the tumor, by even being able to ablate tumor in some scenarios. This is where merging of various research activities and clinical specialties is our strength. We have the ability to do the liquid biopsy that I just mentioned, looking at methylation in the plasma. Andreas Lozano has the expertise on using the high-frequency ultrasound. He is one of the world leaders in this area. And so we put our heads together and said, can we in fact use high-frequency ultrasound to see whether we can enhance the shedding of tumor DNA and other conditions into the plasma, into the bloodstream, that would allow us to more accurately, more effectively diagnose based on the methylation signature in the plasma, the disease condition that we're dealing with, and more importantly, use this non-invasive ultrasound-induced alteration in a tumor or other disease condition to be able to use the blood test in a more effective manner as a screening tool. So that's one example. Another example where a number of specialties come together is the application of chemotherapeutics via angiographic injection of the drugs into tumor cavities. So the work done by Timo Krings and Patty Nicholson, who are two interventional neuroradiologists, they're able to introduce catheters that go to the vasculature that feeds the brain tumor and take advantage of that opportunity to deliver targeted therapeutics into the tumor. And so these would be examples where the crossover of the disciplines can happen under the umbrella of the Krembil Brain Institute. But as I said, you've put me in a difficult spot because there's so many areas that I can go on about, but I know we're limited with time. And thank you for the opportunity to speak.
Well, thank you, Dr. Zadeh, and perhaps impetus for another live stream series in the future. Okay, for our audience, a quick reminder, if you have a question for our scientists, we invite you to go to slido.com, the link's at the bottom of the page. Type in hashtag 424160. You can submit and vote up questions. We'll get to them a little later in the broadcast. All right, time to introduce you to three research fellows, all rising stars at the Krembil Brain Institute. And first, a bit of background about fellows. These are scientists who have completed their medical degrees and residencies and are selected to train in a highly complex subspecialty here at the Institute. They learn additional skills while also advancing the research in the labs and support in the labs the work of lead investigators such as Dr. Weaver and Dr. Zadeh. Research fellowships add to the richness of the educational environment. As well, they create a ripple effect way beyond UHN. That's because many of our fellows become highly sought after to take on research and leadership positions across Canada and around the world. The sum of all this, of course, is UHN is having an impact, a profound impact, delivering on its vision of a healthier world for all. Fellows, by the way, also have the opportunity to pursue innovative research projects of their own, and that's what we want to focus on today. First, we have Dr. Olga Rojas, an immunologist whose research focus is on neuroinflammation in diseases of the brain, such as multiple sclerosis. Dr. Rojas obtained her MD and PhD in Colombia. She's now pursuing postdoctoral studies at the University of Toronto. She joined the Krembil Brain Institute in 2020. Welcome, Dr. Rojas.
Hi, thanks for having me.
It's a pleasure to have you, and thank you for taking the time. I know you're very busy. Your pioneering research is advancing our understanding of diseases of the brain, such as multiple sclerosis, also Alzheimer's and Parkinson's, I understand. And what's fascinating to me is you focus on a cell in the stomach that somehow ends up in the brain. So first of all, maybe talk to us about this B cell. What's the function of the B cell in our bodies?
Yes, you are right, Christian. So my focus is mainly on B cells. So I can say that B cells are present in our body to protect us. So imagine they are pretty much like police officers in our body, and they have shields. And the way the shield works is that they produce substances that we call antibodies. So the shield is pretty much the antibodies. Just to give you an example that we are all living right now due to the pandemic is COVID vaccines. So one of the main reasons we want everyone to get the COVID vaccine is because by getting the vaccine, we will activate those little police officers. They will produce antibodies to protect us against severe COVID disease. And those antibodies are produced mainly by the B cells I am working with.
Okay, so what have you discovered about B cells and what they're doing in the brains of patients, say, for example, those with multiple sclerosis?
Yes, so we found that other than antibodies, for years we have been thinking that the main role of B cells were producing antibodies, but we found that they can produce other substances that can decrease damage in organs like lungs and kidneys, for example. But maybe you know that the brain it's special. The brain has like a big wall around to protect it. So for many, many years, we thought that there was no way that B cells or any type of cell was crossing that wall to get into the brain. Recently, I was able to find some B cells that were derived, as you mentioned before, from the gut. So they were developing initially in the gut, and then we find them in the brain. I still remember the first time we saw them in the brain, and we were like, wow, is this really happening? So we had to convince ourselves and convince all the scientific field. And we now know that those B cells can get into the brain in multiple sclerosis and can help to control the damage of the brain. And now I am working on trying to develop and trying to understand a few more roles of those B cells in the brain. And that will be like the first step to see how we can help the development of new treatments for brain diseases.
And do we know, Dr. Rojas, what triggers the B cells to go to the brain?
Yeah, the magic of those cells that I discovered is that they are in the gut, and they talk basically every second with the bacteria. So in the gut, we have live bacteria. They are living with us. And we know that those B cells talk with the bacteria. Depending on the tone of that conversation, the bacteria can make those B cells to behave sometimes as bad guys and sometimes as good guys. And depending on that, they will migrate to the brain, and they will control the damage of the brain in multiple sclerosis, for example. So there are still so many things that we don't know from those cells. Right now, I am trying to understand what's the type of communication between the bacteria and the B cells in the gut that triggers them to just travel from there to the brain. And which is really interesting is that that will help us maybe to generate new treatments that might be easier. Instead of trying to find something that will access the brain directly, we can drive the communication of the B cells in the gut and see how we can move them the way we want to the brain into basically in different neurodegenerative diseases.
Okay, so talk to us then about the trajectory of your research. Where do you see this going, Dr. Rojas?
So I have to say that my story is a little bit long. I started as my MD program back in my country. I was trained to help patients, but at some point I realized that if I was able to understand what was behind the patient and going a little bit deeper into the science, maybe that will help me to help better. So I decided to do a PhD. During my PhD, I was trying to understand how those little police officers, that means our B cells, were helping us to protect children from a lethal gastrointestinal infection. And that's how I learned about the gut. Then I decided to learn a little bit more about preclinical studies, and I moved here to Toronto, and I started my postdoctoral fellow at the University of Toronto working on multiple sclerosis. And when I was there, I found that everyone was looking at the brain. And I was like, no, wait, there are more things that are interacting with the brain. What could happen in the gut during multiple sclerosis? And that's how I decided to go down and try to see what was happening with those little police officers in the gut. And that's how we found that they were there. And somehow at some point during the disease, they were moving into the brain, and we started finding them there. So for years, we thought that since the neurodegenerative diseases target basically the brain, everything was happening in the brain. The way I see it is that maybe there are more things outside the brain that can interact and impact neurodegenerative conditions. And the thing that I want to focus on is what is happening in the gut, what is the type of communication that is happening between the B cells and the gut for them to at some point maybe go into the brain during some specific conditions. And ultimately, this will help us to develop new treatments that maybe we can start from outside the brain and maybe also at early time points of the conditions.
Okay. You mentioned Colombia, your home country. It was just a few years ago, you left your native Colombia to start a new chapter in terms of your career, and you joined the Krembil Brain Institute last year. What motivated you to take this huge leap in life to come and to be at this institute?
So after all my training, finally I felt, okay, I'm ready to do what I want, which is basically help patients. And I found Krembil the best place for me. And the reason of that is because I felt that at KBI, I could blend myself into an environment full of clinicians, scientists with expertise in neuroscience, myself, which was like a new topic linked with neuroscience, coming from the expertise in the gut. But I also have to say that one of the beauties of joining KBE was that I found that the patients were also really active and participating with us, trying to better understand diseases and finding new treatments. And I thought that was really captivating. It was not only me, it was not only clinicians, it was like a big team effort working together towards our goals. And I thought that will be the best way for me to go, to be able to merge my expertise as an MD, my expertise as a basic scientist, and the interaction with patients to find new treatments for basically Alzheimer's disease and Parkinson's disease. And lastly, I also have to say that I am coming from South America, which is a less resource community in terms of science, and also maybe healthcare. So by the end, joining KBI will help me to give back to the community, not only to the patients. It will help me to give back to the community here in Toronto, but it will also hopefully help me to give back to the community back in my country.
Well, and that speaks to the ripple effect. So thank you, Dr. Rojas. Next up, Dr. Luca Milosevic, whose efforts in deep brain stimulation are aimed at discovering more targeted therapies. Dr. Milosevic received his PhD in biomedical engineering from the University of Toronto, followed by a postdoctoral fellowship at the University of Tübingen's Institute of Neuromodulation and Neurotechnology in Germany. In 2020, Dr. Milosevic returned to Toronto to join the Krembil Brain Institute. Welcome, Dr. Milosevic.
Hi, thanks for having me.
Okay, so your clinical and research focus are in the field of deep brain stimulation, something not all of us are familiar with. Could you start maybe with a bit of a primer to help us understand what is it, how does it benefit patients?
Yeah, for sure. So deep brain stimulation is a surgical procedure which involves the permanent implantation of very small electrodes deep within the brain. And a stimulator that's implanted underneath the skin of the chest is used to deliver electrical impulses to the electrodes and thereby to the brain. And this whole system is, you can kind of think of it as a cardiac pacemaker, but it's essentially a pacemaker for the brain. And the goal of the stimulation is to attempt to correct altered brain activity that we know to be associated with particular diseases. So probably the most well-known and most effective use of DBS is for Parkinson's disease. And it can have really remarkable effects. It can help a patient get out of bed without assistance, or it can suppress their tremors so they can have a sip of their coffee. So as you can imagine, it can have a really remarkable effect on their quality of life.
And I understand it's been used in cases for people with Alzheimer's, dementia, anorexia, eating disorders, depression. The list seems to be growing. And UHN's Toronto Western Hospital is a world leader in doing these surgeries, correct? It performs about 150 a year, you were saying in our pre-interview. And I understand that's where you are intimately involved in terms of these surgeries. Talk to us about your role in the OR.
Sure, yeah. So in the operating room, I sort of help the neurosurgical team navigate to the optimal position within the brain to implant our electrodes. So we're often targeting areas that can be smaller than a cubic centimeter. So what we do is we perform brain recordings to confirm our final implantation location. And this involves the advancement of very, very, very tiny microelectrodes, which we use to record the activity of individual neurons along our surgical trajectories. And these neurons are continuously active. They're communicating with one another, or they're sending signals to control muscles. And essentially, neurons within each region of the brain have distinct patterns of activity. So they essentially speak a different language from one another. So my role in the operating room as an intraoperative neurophysiologist, essentially I'm trained to understand the language spoken by all these different regions of the brain. And we use these recordings to pinpoint the exact location of the structure that we're targeting for our therapy. Now, the other thing that I didn't mention is that the procedures are done generally while the patient is awake. And this is necessary because under anesthesia, all the neuronal activity shuts down. So it's hard to do this brain mapping procedure. But also we need to be able to deliver test stimulation in the operating room to as sort of like a third step to ensure that our electrodes are implanted in the right place. So at my workstation in the operating room, I also have the honor of turning on the stimulation for the patient for the very first time. So when we know we found a good spot, we'll often ask the patient, if the patient has tremor, to maybe stick their arm out or do something that may elicit their tremor. And I get the privilege of pressing a button to immediately stop their tremor. And it's a remarkable feeling. Our surgeons will often ask the patients, when was the last time you've experienced that? And typically our patients can pinpoint an exact date, 10 or 15 or 20 years ago that, they were symptom free. So really, really rewarding to be able to be a part of that.
Yeah, definitely. That's a game changer. You've made it your research mission. You mentioned all these neurons. I understand there's 4 billion of them in the brain. So in terms of your research, talk to us about the potential here. How do you leverage what you're trying to understand in terms of benefit to the patients and the role of the neurons?
Yeah, so of course, I'm in the operating room, so we leverage this opportunity to perform research. So we engage these patients in appropriate tasks. And one of my main goals has been to basically better understand how deep brain stimulation affects the brain. So my background is in biomedical engineering, as you mentioned, And prior to the start of my graduate studies, I hadn't even heard about deep brain stimulation, but I was quickly introduced to it. And after seeing the remarkable effects it can have as an engineer, my first question was, well, like, that's incredible, but how does it work? And I was asked a bunch of, clinicians, scientists, started reading into literature, and quickly came to realize that there wasn't an exact answer to that question. So exactly how the brain activity was being changed by the stimulation was still a mystery. And even though DBS has been around for close to 25 years now, its evolution beyond Parkinson's disease has been rather slow. You mentioned that there are these new and emerging DBS indications, and the clinical trials have been mixed. So to me, I feel that the success of the therapy ultimately comes down to having a very astute understanding of how exactly we're changing the brain activity at the site of stimulation. Of course, with every disease, we're targeting a different region of the brain. And we've done numerous studies along these lines now, so we've been able to describe the exact changes to brain activity that are necessary to abolish a patient's tremor. And we've also been able to describe very, very precise mechanisms that underlie benefit of deep brain stimulation for Parkinson's disease and epilepsy. And after accumulating lots of data while stimulating in different structures of the brain, one of the most interesting findings has been that every single brain region actually responds differently to stimulation. But we've discovered that there are distinct properties of the brain, anatomical properties and physiological properties, that can actually allow us to predict exactly how each individual region is going to respond to stimulation. And in fact, we can precisely tune our stimulation settings to manipulate the brain activity and have it behave exactly the way we want it, or in a functionally relevant way. So we've now described this, and we've actually recently developed a computational model that can simulate the brain's response to stimulation in many different brain regions and at any given stimulation settings. So we're finally getting a more comprehensive understanding of how DBS works. But more importantly, we're hoping that we can use the tool we've developed essentially to develop new strategies to change brain activity in a therapeutic manner to treat neurological disorders that are not yet adequately managed, like Alzheimer's disease, for example.
Well, I was going to say, so the potential here is with your research of actually expanding the use of DBS?
Yeah, definitely the greater goal. I'd also like to just quickly note that, this is just sort of one of the ongoing projects and areas of focus in my lab, but we're also leveraging sort of new technologies, which are allowing us for the first time to wirelessly record brain activity from patients who have sort of these new generation DBS devices. And by means of wirelessly recording activity on demand in the context of Parkinson's disease and essential tremor and treatment-resistant depression, or epilepsy or Alzheimer's disease, we're going to be able to learn so much more about the exact changes in the brain that lead to basically the development of symptoms in these different diseases. And of course, our ultimate goal is to develop the most appropriate and most effective methods to tune or correct this brain activity in a therapeutic manner.
It's amazing. I have to ask, you're advancing your career quite nicely in Europe, and then in the middle of a pandemic, you decide to come and join the Krembil Brain Institute. So what was behind that decision?
When the opportunity arose to come work at the most well-renowned DBS center in the world, it was really a no-brainer. So Toronto Western Hospital and Krembil Brain Institute are often regarded as the epicenter of the modern era of deep brain stimulation. And I have the privilege of working with exceptionally talented surgeons and neurologists and physiologists and basic scientists. We have access to incredible resources. We have a high volume of surgeries. As Dr. Rojas mentioned, our patients are very well engaged. I also hold a professorship at the University of Toronto in biomedical engineering, so I'm able to recruit exceptionally talented graduate studies to come join our program. Basically, everybody involved in this process is always eager to push the boundaries, is dedicated to the highest quality research, and most importantly, improving the lives of patients. So, there was no question about coming here. It also happens to be, where I grew up and I call home. So very, very proud to be in a position to be able to drive this type of innovation forward here.
Well, thanks so much, Dr. Milosevic.
Thank you.
Next, let's bring in Dr. Gabor Kovacs. Dr. Kovacs completed his medical training in Budapest, where he rose to the head of the Department of Neuropathology at the National Institute of Psychiatry and Neurology. From there, he moved on to the Institute of Neurology at the Medical University of Vienna. He's trained at the Indiana University in the US and was a visiting professor at the University of Pennsylvania. Dr. Kovacs specializes in neurology and neuropathology with a research focus on neurodegenerative diseases. He joined the Krembil Brain Institute in 2019. Welcome, Dr. Kovacs.
Welcome, and it's an honor to be with you all.
Your research program zeroes in on the presence of certain proteins that appear in the brains of people with neurodegenerative diseases. Talk to us about your research and the significance of this connection with the protein.
Yes, thank you. So first of all, neurodegenerative diseases are a group of conditions which are characterized by progressive loss of nerve cells in the brain, and also the progressive loss of function of these nerve cells. And nerve cells are the working units of our brain. So obviously, if the function and the network is lost, their communication between each other is lost, then that causes clinical symptoms. Now, 100 years ago, when colleagues at that time used the first microscopes and looked at histological preparates from individuals who died in neurodegenerative diseases, found very strange structures in the brains. And then they developed sophisticated stainings, like using silver stainings. And they saw that in the diseased patients, there are something strange stained with, for example, silver, what they did not see in individuals who did not have this progressive function loss of nerve cells and clinical symptoms. And then we had to wait a couple of decades. And in the early 90s, they found that these strange structures described 100 years ago are indeed composed of proteins. And these proteins are also found in every individual, every normal individual. But in neurodegenerative diseases, these proteins become bad proteins. So the good protein becomes a bad protein. And these bad proteins accumulate in nerve cells. And this leads to a problem, a function loss of these nerve cells. Now, why is this important? Because identifying the proteins as a central in the mechanism of disease allowed researchers to develop methods to detect these proteins associated with disease. So we call these biomarkers, markers of disease, which can be used as a diagnostic tool to diagnose neurodegenerative diseases. And also identifying the central role of proteins led to new therapy targets, either therapies which target these proteins or therapies which target the process which changes the good to bad. So that is my focus.
And a great illustration, if we can dive right into your work, is a 2020 study, I understand, of a neurodegenerative disease called progressive supranuclear palsy, or PSP. Let's just start with, what is PSP?
Yes, so PSP is the abbreviation for a condition called progressive supranuclear palsy. And this is a relatively uncommon, so not so frequent, brain disorder that causes problems with walking, balance, eye movements, and later also with swallowing. However, some patients start with changes in the behavior or speech problems, and some of the patients look clinically like a Parkinson disease patient, but PSP unfortunately is not so good prognosis. So usually the patients, there is no therapy, and it's much more rapid progression than other neurodegenerative diseases.
And talk to us about what you discovered that in your 2020 publication.
Yes, so first of all, other studies found that the protein which is accumulating in PSP brains is called tau. this is, as I said, found in normal individuals, but it is changed in PSP to a bad tau. And what we discovered is that actually there is a spot, a brain region, a region in the brain where the disease starts. So what we found is that irrespective of which clinical symptom predominates in these patients, the disease starts in a small part of the brain. And then the reason why these patients have different clinical symptoms and different progression and disease duration is how this brain region where tau accumulates first, how the tau protein will spread to other brain regions. And we also found that not only the nerve cells are involved in this process, but there is a cell type which we call supporting cells or glial cells, which are very important in the propagation of this bad protein to further brain regions. So in our study, we identified the first place, the most vulnerable area of the brain where the disease starts, and we defined 6 stages, 1 to 6 stage, how the disease progresses.
So talk to us, Dr. Kovacs, how this discovery might be applied one day to help patients with PSP.
Yes, so thank you. So this discovery, first of all, it helps researchers. So we provided ideas, first of all, that we identify this first vulnerable spot. Now, the question is that what is the unique feature of the nerve cells in this vulnerable spot, which makes them vulnerable for this disease? So with this, we provided ideas for researchers to provide in this group of nerve cells and try to find out why are they so vulnerable and try to focus on therapy targets that support this group of nerve cells. Then the second, we identify these supporting cells, glial cells, that are very important also. And this was underappreciated. Nobody cared about this. Everybody was talking about nerve cells that are important. Glial cells are also important for disease. So with this, we provided ideas to develop therapy targets supporting the supporters. So the supporting tissue supports the nerve cells, and we say that support the supporters, and then the nerve cells will live longer. And then finally, we define stages of disease. And what does this mean? This means that the disease starts in a place and maybe the patient does not yet have a symptom. We say that this is stage 1. And this means that there are patients who are in a preclinical stage, which means that if we have a protective therapy and we know how to detect in life this early stage, then we can offer protection to the nerve cells and prolong the time when the first symptoms will appear.
We are getting pressed for time, but I do want to know, Dr. Kovacs, you had a very successful career going in Europe, and you decide to uproot your career, your family, and come all the way across the pond, also to the Krembil Brain Institute. Can you give us a sense of what was behind that decision for you?
Okay, thank you. So I will be short. So let me go back 5 years. In 2014, I was flying from the western part of Canada from a conference, transit in Toronto, landing in Toronto, and my 6th sense, or maybe 7th, I don't know, said in my heart that this is an important place for me. So 5 years later, I met Dr. Anthony Lang, who is a world-famous best neurologist and leader of the Movement Disorders Clinic in Toronto Western Hospital. And he approached me and asked whether I would have want to visit them and eventually work here. So I met him. I saw the colleagues, I saw the building, I saw the facility, I saw a tremendous accumulation of excellent researchers. And the good was that they communicated and they supported each other, and aspects so much underappreciated. The sources were great. And I don't think I hesitated too much. I felt the 6th or 7th sense told me that this is my fate. And I think that lets me talk also about COVID. just think how rapidly vaccine was developed for COVID, because every government said we need that mass effect, a lot of researchers focusing on vaccine development. Why don't we say neurodegeneration is a priority, and then the mass effect, which we have in Krembil, will induce the same results? Thank you.
Thank you, Dr. Kovacs. Now, before we get to your questions, I want to bring in Dr. Zadeh and Dr. Weaver briefly to talk about a couple of things. When you hear the caliber of the work being generated by Drs. Rojas, Milosevic, Kovacs, and the fact that all three chose to come here and do their, perform their discoveries here at the Krembil Brain Institute, what does that say to you?
It says a lot, but just before I begin, I'm just going to add a point of clarification that these three individuals are what we call PIs, principal investigators. They're not fellows. I'll just correct that particular statement. And to me, individuals like this represent the future. They represent where KBI is going. They are bringing their considerable talents and abilities. In fact, I am the one who's honored that they are with us because, as I said, they are going to be moving us in the right direction in all of their areas. And so what does it say? It says that KBI is a lucky place because we can attract such people and such talent.
And Dr. Zadeh, if I could turn to you, talk to us about the role of philanthropy in helping to facilitate all this innovative, forward-thinking research at the KBI.
I would say, Christian, in general, majority of the work that we do that's cutting edge, that's unique, that's niche, is funded and supported by philanthropy. Majority of our research that comes through peer-reviewed granting and funds is very safe, typically research that's already in part been demonstrated feasible. And it's often the very niche and very high risk that makes the discoveries possible, opens doors for us to look at questions in a very different light and to be able to integrate elements that perhaps are not safe, but they're high risk, however, they're high reward. And that's where philanthropy makes a huge impact. And one of the greatest strengths of UHN, and I can say this with confidence, having gone to many institutions across North America and elsewhere in the world, we really do dominate with the amount of support that we receive from grateful patients, from philanthropy in general. And so it puts us at the cutting edge and provides a unique ecosystem where each of us have strengths due to peer-reviewed work that we do, due to the excellence in care that we deliver. But really then we can take it to the next level with some of the opportunities created by philanthropy. So that's where I think probably we have the biggest distinction. And as an example, we would not have been able to recruit the scientists who you heard from in the last half hour without the support of philanthropy. We wouldn't have been able to have the opportunity to have Gabor, to have Luca, to have Olga here, had there not been the environment created to support their recruitment through philanthropy.
Thanks, Dr. Zadeh. Look, we're approaching top of the hour, but we want to get to your questions. I'm just going to quickly go across the board here and ask, can all of you stick around for at least a few minutes to field some questions for our audience? And we want to go to our Slido questions. Top of the heap here is from a Mr. Mark Cremble. KBI is unique in that it is truly a combination of basic and clinical research. Please explain how this is an advantage when looking for therapeutic solutions. Who wants to take that?
Oh, I'll start. It mentions basic science and clinical research, and there's something in between that, translational research. Translational research is what takes the ideas that come from basic science and translates it into products that will be implemented in the clinic and help people. And to have the environment that nurtures this sort of discoveries to happen, you need the basic scientists and the clinicians talking to each other, interacting with each other. You need them cohabiting. We have that environment, and it is this sort of environment which enables us to achieve this. We're not like a routine university. We're not like a routine hospital. We're sort of a hybrid between the two organizations, and this hybrid is our strength.
Next question from Mark Krembil as well. How important are clinical trials at KBI? Dr. Zadeh, do you want to take that one?
Sure. I mean, I think clinical trials are happening at Krembil Brain Institute on an ongoing basis. Our goal is to create a clinical trials unit where all of our efforts are consolidated, where the resources are put together so that we can in fact catalyze and move the clinical trials through our institution at a higher speed so that the turnaround of delivery and implementation of clinical trials and lessons learned from the trials are expedited. And then as in so doing, we can also create a hub and an ecosystem where we can learn from each other's trials. Currently, when clinical trials happen in isolation, discoveries are often not necessarily cross-talking. And by creating this hub and an environment where the ecosystem allows crosstalk between the various clinical trials and learning from one platform to the next will actually exponentially change our understanding of disease. And so the example that I gave for combining high-frequency ultrasound, which other institutions do have access to that technology, but they don't have the strength that we do with combining it with the liquid biopsy. And being able to create the environment that facilitates and really catalyzes in a more genuine manner this degree of interaction, and being able to then conduct trials and learn from each other in the trial designs is what will strengthen KBI to a different level and catapult us to where we need to be for translational application of discoveries.
Next question from Barb. What can one do to prevent dementia or Alzheimer's? Does one's diet matter? Dr. Weaver, would that be you?
Sure, I'll take it on. And Dr. Kovacs can correct me if I make any errors. In the summer of 2020, in the medical journal, The Lancet, published a paper outlining the risk factors for Alzheimer's disease and dementia. And this is something which is actually readily available through an internet search. And they certainly identified factors such as diabetes, repetitive head trauma, and other factors which can increase your likelihood of getting Alzheimer's disease, there being risk factors. Does diet matter? Well, certainly obesity is a risk factor, and diabetes is a risk factor. So in that regard, it does. Is there a particular type of nutrient or food which is preventative? No. If there were, I'd be living exclusively on it as my diet. But I think that protecting your head and keeping yourself stimulated are the best you can do right now.
Next question. I think Luca Milosevic, I think this might be of interest to you. You've expressed an interest in Elon Musk. How far do you think Elon Musk's vision of a brain tech interface is from being real?
I'll stick to a very, very positive answer. First of all, I love that Elon Musk is bringing this world of, neurotechnology to the public. We know he has a very active Twitter account. He's advertising this stuff heavily. And I think that's fantastic for neuroscience in general. But how far are we from, a brain tech interface being real? Well, we've been implanting patients with devices that can completely wirelessly record their brain activity for a couple of months now. And in fact, technologies have been around for the last 2-3 years already. So we're living in this era already. It's sort of just the beginning, but some of the stuff that is perhaps being brought to the public right now is maybe even a few years old. So we're there now. That's where I'll leave it.
Thank you, sir. Next question from Mark Krembil. Aducanumab was approved by the FDA. Please comment on how you think this will impact the Alzheimer's community and neurodegenerative research in general. Who wants to take that one?
Well, hearing silence, that will be my cue. So aducanumab, its approval was conditional and certainly controversial. Certainly a scientific review panel that preliminarily evaluated it, in fact, suggested that it not be approved. And so there's lots of controversy around this particular approval. And certainly this is called a biologic. This is an agent which does have some side effects associated with its use and would have to be taken in large doses very early in the course of the Alzheimer's process, probably even before individuals are symptomatic. So I think that the story of this approval process is still being written. As in terms of its impact, although I do think that this will definitely have impact, it has shone a spotlight on the issues around drug approvals for Alzheimer's disease and the hurdles and the challenges associated with this. And I think that it will bring additional resources and enthusiasm into the area. So whereas I don't think it's going to have widespread distribution in the short term just because of the extreme cost and difficulties with its implementation, I think that overall the side benefits will be there in helping to continue to motivate and support neurodegenerative research in general.
Thanks, Dr. Weaver. Next question. It sounds like Dr. Rojas' B-cell study could have implications for diet. If what is in our gut eventually travels to the brain, what does that mean for food?
Yeah, so it's an interesting question. It's pretty much one of the things that we are trying to address. I have to say that the food has been described as one of the factors that can change what is in the gut. So depending on what we eat or depending on some of our lifestyle, the bacteria that we have in the gut will change. I can tell you for sure that for multiple sclerosis, there are already some bacteria that are described that will increase in our gut to increase susceptibility to have multiple sclerosis. Our goal now is to better understand that for neurodegenerative diseases because that field is less known. But what we also know now is that there is communication between what we have and what is happening in the gut and with cells. And that communication will send cells and also signals to the brain to have an impact. It could be a positive or a negative impact. And those are the factors that we are aiming to understand to drive maybe some therapeutics in the future that will change something in our gut to help our brain.
Thanks so much. And listen, we could go on. We've gone past our time. I want to thank everybody for your patience, for staying on with us. That will have to be our final question. I want to thank all our guests today, Dr. Donald Weaver and Dr. Gelareh Zadeh, co-directors of the Krembil Brain Institute, and PIs, Dr. Olga Rojas, Dr. Luca Milosevic, and Dr. Gabor Kovacs. Thank you all so much for taking the time out of your busy days to be with us today and share your work. To all our friends in the audience, all the Discovery Ball supporters, a big thank you from the UHN Foundation for your continued support. And a reminder, we're hosting 2 more virtual events as part of our Krembil Discovery Series. Next Tuesday, June 15th at 6, we'll be showcasing the Schroeder Arthritis Institute. And the following Tuesday, June 22nd at 5:30, it's the amazing work at the Donald K. Johnson Eye Institute. For more on these events, a recording of today's session, and information on ways to give, keep an eye on your email inbox. In the meantime, thanks for tuning in, everyone. We look forward to seeing you again. Stay safe.